Hospital Clínica Bíblica desde más o menos hace 8 años y llevo 3 años estando encargado de lo que es el ecosistema de I+D+I del hospital. Les menciono esto porque la institución es muy vieja, tiene 93 años de existir y pese a estar en las ciencias de la vida, en este ámbito, pues haber abierto un centro para la innovación, un instituto de investigación biomédica, ha llevado un cambio de cultura porque no es usual. Entonces, hoy nos enorgullece muchísimo tenerlos a todos ustedes, tener a la doctora Izrael, y también nos enorgullece mucho ser parte de CR Biomed, que es la primera vez que podemos ya participar como miembros y estamos muy contentos en lo que podemos hacer en conjunto para poder llevar a Costa Rica y a nuestro ecosistema al lugar donde siempre creemos que puede estar. Tenemos muchísimo talento nacional acá y la idea es poderlo potenciar para que podamos llevar esas soluciones a los pacientes, que se puedan impactar de manera positiva sus vidas, cambiarles esa calidad de vida y resolver muchos de los problemas que tenemos hoy en día en nuestro ámbito. Entonces, si nosotros podemos seguir promocionando, podemos seguir apoyando este tipo de actividades, pues será un placer para nosotros. Entonces les agradezco mucho a los miembros de CR Biomed por habernos dejado participar, por organizar una charla de la doctora Izrael, que estamos muy emocionados en recibirla, y doña Patricia, le cedo la palabra. Mucho gusto. Les tenemos también un café, un poco de té atrás y unas galletas para los que quieran, y cualquier duda o consulta estamos por aquí atrás.
Bueno, muy buenas tardes a todos y muchas gracias por estar aquí con nosotros esta tarde, y sobre todo a la doctora Israel que nos está acompañando el día de hoy, y también al doctor José Camacho, quien muy amablemente nos ha permitido estar en esta sede el día de hoy, que va muy de acorde con la charla que vamos a tener. Quiero, en nombre de CR Biomed, el Clúster de Biotecnología de Costa Rica, darles la bienvenida, y quería comentarles muy rápidamente de qué se trata el clúster. El clúster se formó en el 2015-16, aproximadamente llevamos unos 7 años. El clúster es como su nombre lo dice, es una agrupación que tiene que ver con diferentes ecosistemas para tratar de hacer el ecosistema de la biotecnología en Costa Rica una fuerza. Está compuesto de 5 áreas. Una tiene que ver con el área de la academia, el área de la empresa privada, el área de gobierno, lo que son los investigadores que no pertenecen aún a una empresa privada, pero que sí están muy llenos y enfocados a la parte investigativa, y finalmente los que hacen el fondeo. Entonces nuestra organización está compuesta por esas 5 diferentes tipos de grupos. ¿Y en qué? En biotecnología en general, la biotecnología de todos los colores, ¿verdad? Hoy estamos específicamente en un tema que tiene que ver de la biotecnología roja, humana específicamente, pero de igual manera tocamos temas de biotecnología roja veterinaria o biotecnología verde, que tiene que ver con toda la parte agro, que Costa Rica tiene importantes fuentes y materia prima para trabajar con eso. Lo que es la parte de nanotecnología, el Centro Nacional de Nanotecnología en Costa Rica es parte del clúster, donde se trabaja más que todo con temas de materiales, todo lo que tiene que ver también con la tecnología y biotecnología de reutilización de residuos agropecuarios, y aquí en Costa Rica de nuevo es un lugar que tiene muchísima oportunidad para hacer ese aprovechamiento de esas materias primas. Voy a pasarme un momentito en inglés. Como ustedes saben, la charla va a ser en inglés, tal como lo habíamos comentado, para ahora mencionar y darle la bienvenida a la doctora. So, thank you for joining us today. It is very nice to have you here. Doctora Israel was actually born here in Costa Rica in this hospital, so we're glad to have her come back. and she has family here, and it just coincided along with the efforts of Peripotent and our offices in Peripotent in Israel, that they spotted her, and she can join us today to help us with cell therapy and tell us the advances and her experiences in that area. So now I'm going to pass the word to Dr. Christian Marin, who pretty much is, we're happy that he's part of our member of our cluster, and I think everybody knows Christian. He needs pretty much no introduction, and he will be introducing formally Dr. Israel. Okay, so thank you.
Thank you very much. I have the great honor of introducing Dr. Israel. She holds a PhD from the Weizmann Institute of Science and is an expert in the field of stem cells and cell therapy. She has over 20 papers and patents in this field and is committed to developing these incredibly novel treatments into things that can really help patients in the future. She's developed products for the treatment of neurological diseases like ALS and insulin-dependent diabetes. I'm really eager to hear about these things that she's trying to take towards the clinic. So thank you very much, Doctor. A real pleasure.
Okay, thank you all for the kind invitation. It's great to be here. And as all said, I was born here in this Clinica Biblica. I won't say how long ago, but it was a few years ago. And I'm very, very pleased to be here and share what we learned in the field of stem cells, cell therapy, and share with you what was done in the field. So this is a short disclaimer saying that I am from a company called Kadima Stem, which is moving forward. In Hebrew, Kadima is moving forward. Stem is stem cells, and we'll talk a little bit about it. So what do we have today in our menu? So first of all, we have a small introduction about what is a regenerative medicine. And then I'm going to speak about lessons that have been learned from our experience in the field of neurology and also diabetes. And finally, I will speak a little bit about what tomorrow holds. So what is regenerative medicine? And I want you to be very open. If you have questions, you can raise your hand during the talk, ask, tell me to speak slowly because sometimes I'm very, very excited and I speak very, very fast, so you can tell me to slow down. So what is regenerative medicine? So these are basically the treatments that are based on producing new cells, target cells, and transplanting them in order to repair our body. And I like to simplify it by saying it's like spare parts. Okay, something goes wrong with your car, I don't know, the wheel or the generator or the radiator, you replace it. So the main idea is to actually generate new cells such as liver cells, lung cells, brain cells, and then transplant it in order to repair the malfunctioning tissue. Okay, so this is the first, let's say, the big goal mark of the regenerative medicine. The second pillar, the second option, is to boost or stimulate endogenous stem cells that reside in our body. Okay, so we have progenitor cells in the brain. We can give some kind of material that will regenerate these cells, and the organ will be regenerated and restore its function. Okay, so we can generate new cells, transplant them, or we can induce the endogenous stem cells to regenerate our organs. Okay, so how can we do it? Okay, so for that aim, we are using stem cells as our starting material to manufacture, to generate cells. And there are several kinds of stem cells. The most naive ones is once you have a fertilization, egg, sperm, meat, you have the potent stem cells, which can give rise to any cell type of the human body, and it can also give rise to the placenta, okay, the supporting tissue that supports the development of the embryo. And it can be expanded or proliferate indefinitely, so you can have a lot of starting material and then push it towards specific organ. The next level is after 2 to 3, 4 days of gestation after fertilization in vitro. we have embryonic stem cells or pluripotent stem cells. Here you have the blastocytes from which the embryo is generated. And we can extract the inner cell mass, which are the cells that are inside. There are about, let's say, thousands of the cells. And they share the same phenotype, the same properties. And they can proliferate. We can expand them indefinitely, okay? And then push them or mimic the embryonic development in order to push them into a specific cell type. Okay, this is the whole principle. So these are the most naive and most robust cells to use them as our starting material. Other layers of stem cells are the multipotent stem cells, which reside in each of our, let's say, derm tissues. For example, we have ectoderm, we have mesoderm, we have endoderm, so we can extract stem cells, multipotent stem cells, and they can expand, but they have limited capacity to expand, and they can differentiate only towards the lineage that they were originated from. Okay, so if we extract mesenchymal stem cells, they can become cartilage, they can become bone, and they can become muscle, but they can become neurons, for example. So they are very limited in their ability to proliferate and also to differentiate. And then the most lower level is the unipotent stem cells, which are the progenitors of specific cell type. For example, neural stem cells, so one neuron can become another one. But again, the proliferation, the extension is very, very limited. So we are using pluripotent stem cells in order to generate our spare parts. So, you see, because so there are two kinds of stem cells or pluripotent stem cells. One is the embryonic stem cells, which I described, which is the fertilized eggs, and you extract the inner cell mass, and then you can expand the cells. And the other one was generated by Shinya Yamanaka, okay, which is called the induced pluripotent stem cells. Are you familiar with this terminology? Induced pluripotent IPS? Okay. So you have two types. One is embryonic stem cells, and the other one is IPS or induced pluripotent stem cells. And as I said before, the embryonic stem cells, you derive it from blastocytes. So you take these kind of cells and you grow them in a tissue culture plate, and then you do whatever you want to do with these cells. Okay? They can become cardiomyocyte, nervous cells, immune system cells, for example, and they can actually be a kind of a triplet. And then you have the induced pluripotent stem cells, which is a terrific invention. Shinya Yamanaka won the Nobel Prize, Nobel Prize winning for generating induced pluripotent stem cells by introducing four transcription factors such as MYC, SOX2, etc. And each kind of somatic cells can become a young cells, can become embryo, such as pluripotent stem cells. Okay, so you can take this cell from your skin, introduce these four factors, and it can be the pluripotent stem cells, very naive cells that you can expand and then differentiate the cells toward any cell type of desire. Okay, so this is the two main principles that I want you to take home. Okay, you have pluripotent stem cells, two sources. One is IPS, induced pluripotent stem cells, and the other one are the embryonic stem cells. So how does it look like? Okay, when you extract these embryonic stem cells or pluripotent stem cells and you grow them in a dish, so it looks like this. This is an example of embryonic stem cells, and this is an example of induced pluripotent stem cells. Here the cells are fibroblasts, which support the cells to grow. And once you want to move to the clinic, you need to get rid of these fibroblasts. So they are xeno-free or animal-free way of culturing these cells. And these are conditions of culture-free media, and you can start as the starting material for generating the target cells of this RNA. So I will walk you in a nutshell on the process of how we develop our products. Okay, so you have an idea. Okay, each one of you work in some kind of a field. You want to generate specific cell type, and for example, you want to do cardiomyocytes for repairing a heart infarction. Okay, so how do you do this? You have an idea. Okay, you say, okay, I want to mimic embryonic development And you take the very naive cells, the pluripotent stem cells, and you differentiate them by mimicking embryonic development toward cardiomyocytes. Okay? So you had an idea. You start with a differentiation protocol. Then you need to show that these cells are working. You need to show that they contract. You need to show that once you transplant them into animal model, they can repair the heart of the animal. Okay? And you need to show the mechanism of action. You need to identify the cells. You need to show that the markers of the cardiomyocytes are the markers that they exhibit. You need to show that they contract in a certain pulses. So this is the first step. And then you write a patent. Okay, this goes to you. Okay, you write a patent and you say, okay, I know I have differentiation protocol. I know how to produce specific cell type. And then you protect your IP, your intellectual property, by submitting a patent. Now, if you want to move forward toward a product, you need to go to advanced preclinical trials. And this means you need to do what is called to tailor the CMC, the chemical, manufacturing, and controls. Meaning you need to show how you grow the cells, which kind of materials you're using, how do you culture the cells, how do you test the cells, quality control test, okay? And everything needs to be according to the regulatory, okay? We usually use the FDA as our regulatory agency that we need to follow, but you can do it locally. Here in Costa Rica, Ministry of Health. In Israel, we did it also using the Israel Ministry of Health. So this is the next step, okay? And then most importantly, before you go to clinical trials, is to show that your product is safe. Okay? Meaning that you're going to inject it to human and it's not going to do any adverse events or be toxic. For God, we didn't kill somebody. Okay, so for that aim, you need to do very comprehensive safety studies. And for that aim, you need to take the right animal model. It can be large animals such monkeys, pigs, mice, and you need to do it accordingly in order to show that your product is safe. And you need to show that all the tests during the way are safe. For example, that you don't have any kind of contaminants in your product, that the product is sterile, and etc. Okay? Once you have all this package, okay, you move to the next step, which is the first clinical study, okay, or the phase 1 to A clinical study, where the primary endpoint, as I said before, is safety. Okay, let's see that you don't harm anybody. And then the next level would be to see if it actually helps somebody. Okay, so this stands for the 2A, the efficacy arm. So if you are succeeded in showing that your product is safe and it managed to do something, you have some kind of a signal or clinical signal, you can move towards the next clinical phases with a larger number of patients using a placebo, etc. And then by the end of the way, fewer years go by, okay, it's a very long process, it takes about a decade from here to here. And then you, if the product is good, you can market, you can get the BLA from the FDA, and then have the product approved. Okay, so after doing all this introduction, I will take you through what we are doing, what we have done in the company that we have founded. So the first vision is to, I call it the 3 R's, okay, replace, restore, repair, malfunctioning tissues, malfunctioning cells, by transplanting new and functional cells. So as I said before, we are starting with the pluripotent stem cells, the very naive cells that can become everything. And then we have developed 2 protocols. One is for the differentiation of these cells toward astrocytes. We call it AstroRx. Astrocytes are the central nervous system supporting cells. Okay, about 30 to 40% of our brain and spinal cord are consistent of astrocytes. It helps you to understand what I'm speaking now. Okay, it supports the neurons. And this is for ALS indications, a motor immune disease. I will elaborate on that later. And it can also be used in other neurological conditions. We started with ALS because it's a devastating disease. The regulatory pathway is faster, and once we prove efficacy there, the way is going to be much easier to go to new indications. And another arm is islets of X, or islets of Langerhans, or the insulin-secreting cells for the treatment of somebody. Diabetes. Thank you, just to see that you remember. So let's start with the neurology, ALS. So ALS, amyotrophic lateral sclerosis, also known as Lou Gehrig disease, from the name of the baseball player, Lou Gehrig. In this disease, the motor neurons are dying. Okay, the upper motor neurons go from the cortex towards our spinal cord, from our spinal cord towards our muscles are dying. So the upper and the lower motor neurons are dying, and this leads eventually to death because you lose muscle function, and then you get paralysis, and then eventually you die. And from onset till death is about 3 to 5 years from diagnosis till death. And about 90 to 95% of the cases are from sporadic with unknown familial background. And 5 to 10% of the cases are with no mutations. And some of the mutations are listed here, such as the C9orf, the SOD1 mutation, the TDP, etc. And there is no cure, okay? No cure, no real effective treatment for ALS. About 450,000 patients are diagnosed with ALS worldwide. And there are 2 FDA-approved drugs, but with very modest benefit, okay? They just prolong the life in about 2 to 3 months. It's no real effective treatment. And one of them is Rilotec, which the mechanism of action is by increasing the endogenous astrocyte ability to uptake glutamate, okay? And another drug is Radicava, which is an antioxidant. And the reason that why I'm saying this, it is because each drug works through one mechanism of action. And once you transplant cells, they work throughout a lot of several mechanisms of actions and have a crosstalk between the central nervous system and the transplanted cells. So it's supposedly it should be with more benefits. So though the motor neurons are dying, it was found that the astrocytes of ALS patients fail to support the neurons. Okay? So they fail to secrete neurotrophic, neuroprotective factors. They fail to reduce toxic factors such as glutamate. Okay? Once glutamate is residing in the synaptic gap, it causes excitotoxicity, and eventually the motor neurons is dying. They fail to regulate oxidative stress and also immune modulate. So the rationale was, okay, we have motor neurons that are dying, we have astrocytes that are not functioning, let's transplant healthy and functional human astrocytes to see if we can restore the function of the endogenous astrocytes. So we moved to the next level, generating human astrocytes from peripotent stem cells. So we have embryonic stem cells, we have a differentiation protocol with neurospheres inside. Then we are generating astrocyte progenitor cells, and then we freeze our astrocytes as an off-the-shelf cell product. So in our next clinical trial, we're going to thaw the cells and inject them directly into the patients. Okay? So this is the process, and very important to say here that from each step, okay, the starting material, the pluripotent stem cells, the APC, the astrocyte progenitor cells, the astrocytes themselves, we can freeze the cells in what we call a GMP, a good manufacturing practice. We have such GMP facility in our company, and we freeze the cells in cell banks, and we have master and working cell banks, and we test the cells through comprehensive tests of quality control, etc. Okay? Okay, so what do we test? Okay, I'm saying we have astrocytes, but you need to believe me. Okay, so how do you do that? Okay, I need to show you that the cells profile have the identity of human astrocytes, that they are pure, there are no residual pluripotent stem cells or other contaminants of other cell types, and that the cells are functional. So here you can see that the astrocytes themselves express astrocytic markers such as GLAST, which is the glutamate transporter, CD44, acylcholine-4, GFP, nestin, and we set up a release criteria for our cell product. In early stages, we set the bar low, and once we accumulate more data, we can increase the bar and say, okay, the cells are not CD44 at 85%, but say 100. But in the first stages, you set the bar relatively lower. And then we show that there are no residual pluripotent stem cells in our culture, meaning there are no 260 SSE4 or EPCAM, which are markers for pluripotent stem cells. So they disappear. So the cells are committed, they are target cells without any residual pluripotent stem cells. And then, of course, you need to do all the tests, the endotoxin, the mycoplasma, very comprehensive viral testing showing that there are no viruses in our culture, and that the culture itself is sterile. And we showed that the cells know how to uptake glutamate from the media. So once we put about 450 micromolar glutamate in the culture, we see it disappears, meaning that the cells know how to uptake the glutamate, so they are functioning. And we show that once we block these transporters, the ability of the astrocytes to uptake glutamate disappears. We show that once we put the astrocytes with neurons, the neurite outgrowth is better. You see here how the neurons in green are thriving, a lot of neuron axons. And we saw that it also repairs once you put oxidative stress, okay, very similar to some of the conditions that are in ALS patients. It actually rescues the performance of the motor neurons. What else? And then we asked, okay, which kind of factors are secreted by our human astrocytes? And we did what is called a secretome analysis using a multiplex. And we saw that about 30 to 50 different factors are secreted by our human astrocytes. And this is a very long list, also it was published. And we have factors such as BDNF, VGDNF, MIDKIND, 604, and etc. Meaning that the cells know to secrete factors that help the survival of the dying motor neurons in ALS disease. Then we move to our in vivo functional assays, transplanting the cells into an animal model of ALS. For example, this is the human superoxide dismutase 1, which is one of the mutations in ALS patients. And we did a transplantation very similar to the route of administration that we're using for humans, meaning intrathecal transplantations through LP, through lumbar puncture procedure, injecting the cells to the cerebrospinal fluid, okay, and then the cells can migrate. They can migrate and reach different parts of the brain, migrate and reach different parts of the spinal cord, affecting systemically the dying motor neurons. Okay, so we monitored the survival, breath, motor, muscle, etc. And we found out that once you transplant human astrocytes intrathecally to the animal model of ALS, it actually delays the onset of the disease, as can be seen here. It prolongs life expectancy. It improves the motor performance of the animals, meaning, for example, there is an assay which is called the grip strength. So the animals are stronger. The rotor road, and I think I have here, I will show you a small movie. If it will, okay. So here you can see two animals. One that was injected with Astrorex, the other one is a control one. Okay, this animal was injected with Astrorex cells. This animal wasn't. It's a sham control. The animal is very weak. It doesn't have muscle strength in its arms and legs, and also the tonus of the tail is very, very weak. So this is a demonstration, in vivo demonstration, that they had a pre-IND meeting with the FDA. Okay, it's a way to correspond with the FDA and to see that all your studies are done by the regulation, and they recommended a study in which we transplanted astral eggs to about 600 NSG. NSG are immune-compromised animals, and just to show that no tumors are formed, no teratomas are formed, and once we inject the cells into the CSF, into the cerebrospinal fluid, they remain in the CNS, they don't migrate to the heart or to the liver. And so for that aim we did this study. And as it can be seen in mice, we inject to the cisterna magna because it's very hard to inject the LP. And we segmented the spinal cord and the brain, and we found the cells attached to the pia mater, very in close proximity to the CNS parenchyma, okay? And they envelope. So for example, this is a section of the cervical region of the spinal cord, and here in red you can see the cells that envelope this spinal cord segment. The cells could be found also in the lumbar, the sacral region, the thoracic, and also in different levels of the brain, meaning once you inject the cells, they migrate through the CNS, and they dock near the CNS parenchyma, and by that extract their therapeutic effect to the dying motor neurons. And it was a 9 months follow-up time. Okay, this was one of the demands of the regulatory agency, is that you will show that after a long period of time, you can detect the cells, and there are no safety concerns which reside from the injected cells. So no tumors will form, no teratomas, and the astrocytes preserved their astrocytic identity. You see, this is a marker for human GFAP, human astrocytic marker, and it can be found enveloping. This is a relatively lower, the lumbar region. So the cells in red can be found enveloping the lumbar spinal cord segment, and some of the bundles that go through the spinal cord. Okay, so after doing all this work and moving from R&D manufacturing conditions to GMP manufacturing conditions, growing the cells in xeno-free conditions and showing all the needed data, we got the approval for the clinical trial, for our phase 1/2A clinical trial, first time done in humans in Jerusalem, in Israel. And then there we injected 5 patients with a dose of 100 million cells, an additional 5 patients with a dose of 250 million cells, just to see that the cells are safe, and maybe we can find some kind of efficacy signal. So in terms of, this was an open label study, in terms of the study design, in order to see whether there is an effect, we measured for clinical signs pre-treatment. Okay, so we follow up before transplantation for 3 months. We measured different parameters, okay, such as the ALS-FRS score, which is the gold standard for measuring the progression of ALS disease. So we have the baseline, so we can anticipate, see if we can stabilize the disease or not. And then we had a follow-up of an entire year just to see if there is any effect and to show that the cells are safe. So in terms of safety, we had no severe adverse events which were related to the treatment, so the cells were found to be safe, with no treatment related to SAEs, nor those limited toxicities. And in terms of efficacy, we were surprised to see that also in cohort A, which received 100 million cells, and also in cohort B, that received 250 million cells, we saw that we can stabilize the disease for a duration of 3 months. Okay, so for example, we have a deterioration of minus 0.88 points per month before treatment, And after treatment, we could have stabilized the disease, which wasn't significantly different from zero, meaning it was stabilized. But after 3 months, the disease progressed. So for our next clinical trial, this one will show, so our next clinical trial, we intend to do repeated dosing, okay? To see if after 3 months we can inject, prolong the effect, 3 months inject, prolong the effect, and see if we can stabilize this disease. So I guess that by the end of this year, we're going to submit our IND package with our off-the-shelf cell product, okay? We are now establishing the clinical protocol for the phase 2B study. It's going to be a placebo control arm. So okay, we're going to have a control arm and also a treatment with about 20 patients in the treatment arm and 10 patients with a control. And there is something which is called REMAT, which is a regenerative medicine advanced therapy. So if you have a disease and you want to have an accelerated regulatory pathway, you can submit the REMAT. And then once approval, you have more interactions with the FDA and all the regulatory pathway is expedited, okay? So keep it in mind once you develop your own cell product. So this is it for our, yeah.
Before you go continue to the next one, how long did it take you from the beginning until the point that you're right now?
It's an excellent question. We started the project when my middle daughter was born. She's already 9 years old. She's here in Costa Rica, so it took us till now 9 years. Yeah, I would call it an advanced therapy. Okay, it's not a molecule. It's not for something, but it's biologics. Okay, so it takes more time. The regulatory learns with us how to deal with such a innovative drug product. Okay, also from the gene arena and also from the cell therapy arena, the regulation learns with us.
Do you have an idea of how much longer it will take?
Yeah, so we have the phase 2 and then the phase, once it will succeed, the phase 3. I hope it will take us between 5 to 7 years till final approval. Okay, because each clinical study is between 2 to 3 years. Okay, so you cannot skip, the hard work.
How long does it take for the cells to integrate into the normal microenvironment?
How long does it take? A few days. After a few days, we already saw them attached to near the CNS parenchyma. Few days, and they stay there for about 9 months. And now we will move to another cell product that we are doing. Okay, so we are trying to see if we can develop a cell therapy for diabetes, insulin-dependent diabetes. For that aim, we need to mimic embryonic developments towards the pancreatic or endodermal lineage. I don't think I need to elaborate about diabetic. But I will say a few facts. First of all, we have about 180 million patients which are insulin dependent worldwide. 45 of them are type 1, the juvenile form, and the rest of them are type 2, which are insulin dependent, 30% of the cases. And it's a burden. It's a burden to the patient, it's a burden to the healthcare. We need to find ways to restore the function of the pancreas or the insulin secreting cells. So we have treatments today. We have very good and effective treatments using sensors which sense the glucose levels in your blood, very reliable ones. There are several companies which are now developing closed-loop systems. And we have the pumps, okay? There are wearables pumps that you can put and then replace every 3 to 4 days. And you can restore very nicely the glucose levels in your blood. But still, it's very, very labor consuming in terms of the patients. There are plenty of episodes of severe and unexpected hypoglycemic episodes and hyperglycemic episodes. And you cannot escape the progressive diabetic complications such as heart diseases, neuropathies, strokes, and etc. So we need to develop not good treatment, but a cure. Okay? So using peripotent stem cells or embryonic stem cells in our starting field, and then having an insulin-secreting cells or islet of Langerhans can be of a very good solution. And a very good proof of concept is the Edmonton protocol. I don't know if you're familiar with it, but there is a protocol which you derive islet cells or insulin-secreting islets from cadaveric donors, from people who died. You need about 2 to 3 people which will donate their pancreas. You extract the islets of Langerhans, and then you transplant the cells, and it can restore the function of the pancreas. Okay, so the patients are normal glycemic, they don't suffer from hyper and hypoglycemic episodes, but you need to give immunosuppression, okay, because it's cadaveric donors, it's not autologous, and the supply is limited. Okay, so these protocols that developed since 2000, and since now about only 200 patients, 2,000 patients were treated with this protocol. So it's a very good proof of concept that if you have very good islets of Langerhans, it actually can cure diabetic, but still there is not enough supply. So our solution is actually to overcome donor tissues availability shortage, replace malfunctioning islets cells with healthy ones. It will maintain the continuous glucose levels in the blood, and find a way to avoid the immune suppression. Okay, and I will elaborate on how there are several ways to avoid it. Okay, so how do we do it? This very similar to what was done to the astrocytes. We mimic the embryonic development, going through endoderm development, going from very naive cells, peripotent stem cells, a 7-stage procedure, moving from definitive endoderm to primitive gut tube, towards the endocrine progenitor cells, immature endocrine, and then we have each ball here is an islet of Langerhans. Okay? So we have insulin-secreting cells. This is a flow cytometry analysis where you can mark the cells and know exactly which percentage of the cells are positive. And C-peptide, you have a pro-insulin once the pro-insulin is cleaved. Okay, you have the C-peptide and the insulin, so you use C-peptide to show that the cells secrete insulin. Okay, this is a marker. So you see very nicely that about 50% of our cells are secreting insulin. And if you want to avoid the immune system, we have a way to encapsulate the islet cells. Okay, so each ball here is an islet which is encapsulated by alginate. Okay, alginate is some kind of hydrogel envelopes that protect the cells from the immune system, but do not disturb sensing of the glucose, and the cells can secrete the insulin to the bloodstream. Okay, this is one of the ideas that we have. Okay, just to show you, this is the characteristic. What each ball that I have shown you here looks like that. This, okay, you have the C-peptide, which is the insulin peptide. Most of the cells secrete insulin, but this is an islet, so you need to have glucagon, there are somatostatin, and other cells that belong to the endocrine system. So the cells are PDX positive, glucagon positive, just to show you that this is a very nice, pure endocrine islet of Langerhans. Okay? And then we envelope the cells using this alginate. What we have developed we found and developed in our lab is that we can even purify even more the cells. Okay, we found that CD49 marker is a marker for insulin secreting cells. So if we have a mix of immature cells, maybe glucagon cells, we can sort them. There is a magnetic way of sorting, picking up the cells, okay, and then we have a pure population, or let's say purer population of insulin secreting cells, making the cluster that we have more efficient. So we can transplant less cluster per kilogram body weight. as previously shown, we need to show that it works in vivo. So this is an STZ animal model that mimics insulin deficiency. And here you can see the control arm. Here it stopped measuring because the animal died. And these animals were injected or transplanted with encapsulated islets of Langerhans, and we restored the glucose levels in these animals. So now in the company, we are still in this program. We are in our preclinical stage, in our advanced one, and we need to find a way to deliver the cells. Okay? So there are some considerations. Okay, first of all, we need to avoid immune rejection. We don't want to give immune suppression because we lose more than we gain. Okay, we need to avoid foreign body response, meaning avoid fibrosis. Sometimes when you inject something, it covers with fibrosis in order to protect the body. Okay. And once it's covered with fibrosis, it cannot sense the glucose levels and the insulin cannot be secreted. Okay, so we need to avoid this one. It should be retrievable. Okay, if something goes wrong, okay, if there is some kind of infection, if for that reason there is some kind of a proliferation or something which is not controlled, we want a way to remove it from the body. Okay, so it should be retrievable. And the first thing, it should be vascularized, okay? Because we put it in the body, we want the bloodstream to go inside or in close proximity to the device, so the cells will sense the glucose levels and secrete the needed amount of insulin. So there are several options, okay? I'm approaching the last part of the talk, so this one, okay. So free islets, this is the ideal solution, okay? But since this is an allogenic transplantation, meaning it doesn't come from autologous, we need to find a way to avoid rejection. So today there are several new lines of pluripotent stem cells which are called hypoimmunogenic, okay? Meaning that the MHCs are removed from these cells, so the body won't recognize it as foreign. And the injection can be won't be rejected. Another way of avoiding the immune system is by clocking, covering the cells with some kind of molecules that make the immune system blind to them. Cancer cells, for example, are doing that. Okay, they're putting PD-L1 antigens on the top of the surface, and then the immune system doesn't see. Okay, so these two ways are now in process. Still in the research grade, but you know, this is the future, okay? Having pluripotent stem cells, hypoimmunogenic, so you can transplant directly, target cells without without the immune system rejecting it. So this is it. But still, we have the retrievability, okay? If we put free islets, we want to retrieve it, okay? It's going to be very hard. Okay. Next solution, such as our own, okay, we encapsulate the islets, okay, so it's not rejected by the immune system. We already seen it using animal models, but again, we cannot retrieve it. Other system is what we call macro encapsulation. What we do is micro encapsulation. Each islet is covered, but there is a micro encapsulation. So there are devices. There are several companies now developing devices in which you can put your islets inside, such as a MailPen, Visai, Beno2, 3D bioprinting. Okay, we can print some kind of device and put cells inside. Okay, so these are solutions that we are now exploring with several collaborations with several of these companies to see if we can protect the cells using these kind of devices. So this is, in a nutshell, the strategies that we are using. One is immune suppression, we're not going there. The other one is microencapsulation, encapsulating the islets of Langerhans. The other one is having what we call universal donors, or hypoimmunogenic or clocked pluripotent stem cells. And the fourth one would be, to have the macro encapsulation, the device itself. So this is it about the second program that we have. what the future holds is that the sky is not the limit. Just some statistic get you more enthusiastic that Costa Rica can be part of this effort in curing diseases. So these are the clinical trials being conducted worldwide using pluripotent stem cells, the embryonic stem cells, derived cells, or the induced pluripotent stem cells ones. So you have here the statistics, and there are only 20 clinical trials, okay, which are interventional, meaning we are transplanting target cells. And most of the clinical trials are done with RPEs, which are the retinal pigmented epithelial cells. Very easy to differentiate, right? Very easy to obtain for the treatment of ocular disease, AMD. And very few, I think only one already with the type 1 diabetes, and we hope to be the next one. And several, one was just recently approved for Parkinson's disease, where they generated dopaminergic neurons. Okay, so it was approved in the US by Neurology company, and our own. So I guess we can add some point here in the future. If one of you would like to, let's say, start something in this direction. The next level, of course, okay, so now we can generate cells. The next level would be to generate tissues. Okay, so there is a 3D printing technologies out there. None of them is in the clinic yet, but you can manufacture heart. And here you need to be smart. Okay, you need to combine different cell types. You need to combine muscle cells with the vascularized cells. So you need to structure the whole organ. But this is the future, okay? So I believe that within 20 to 30 years, we're going to have something in the clinic already using 3D imprinted organs. And this is it. And I will conclude by saying, as Walt Disney said, okay, if you can dream it or imagine it, you just can do it. So this is it. So you can see.